So without further ado, let's get going. As I said, I'll talk about our science mission, JGI programs, our operation as a user facility, and just a word about project management. So JGI is a user facility that provides high-throughput DNA sequencing and analysis in support of DOE missions in alternative energy, carbon cycling, and bioremediation. Pretty succinct. Important elements here are a user facility and sequencing and analysis that support these DOE missions. Well, who are we? We are about 300 FTEs scattered across 5 partner labs. Most of our operation is in Walnut Creek, but we also have, and that in Walnut Creek, our employees come from either Lawrence Berkeley or Lawrence Livermore National Lab, but we also have essential partners that contribute to our programs at Los Alamos, Pacific Northwest National Lab, and at the Hudson Alpha Institute, formerly at Stanford Human Genome Center, but recently moved to Birmingham, Alabama. We produce over 300 gigabases of sequence a year in support of DOE mission-relevant projects, and that's parceled out between over 400 active projects at any one time. So we're a busy organization with a lot to keep track of. JGI Science Mission is driven by the Office of Biological and Environmental Research, and our program manager, Dan Drell, is paying close attention to what I say here to make sure that our efforts to JGI are tightly focused on DOE missions. And those are primarily bioenergy, global carbon cycling, how CO2 moves around the planet, how we might actually capture it more effectively, and biogeochemistry, a term that has recently replaced the term bioremediation, which has fallen out of favor. Biogeochemistry refers to the movement of toxic materials and their capture or degradation in the environment. Now, JGI's core products, you see down here at the bottom, are plant genomes, microbial genomes, both prokaryotic and eukaryotic microbes, and metagenomes. It's useful to consider how these 3 mission areas tie to these various core products of the JGI. If you think about it, in the critical issues in bioenergy are how do we create enough biomass that we can subsequently use to produce fuels without altering or without consuming all of the arable land in the U.S. and crowding out food, the production of food. So understanding how plants work, how to get more biomass onto an acre of land, or how you might modify the plant cell wall in such a way that it's more readily degraded so that you can extract more sugar from the plant cell wall, critically important elements of the bioenergy mission. The microbes in the environment that have figured out how to degrade the plant cell wall, and we need to understand those processes by understanding the microbes that carry out that process. And likewise, we have a very active program in the metagenomics of microbial consortia that are responsible for degrading biomass. So each of these core areas contribute to the bioenergy mission. Likewise, for carbon cycling and biogeochemistry, there are plants that as major photosynthetic organisms and as sequesterers of toxins from the environment contribute to carbon cycling and biogeochemistry. Likewise, microbes and metagenome projects are easily fit into these mission areas. So in each of the mission areas, we are carrying out projects in each of our core areas. And the overall strategic vision for the science programs at JGI are to be able to contribute genomics of scale, things that individual investigators cannot easily do by themselves, that accelerate our response to energy and environmental challenges, which you don't have to do more than pick up the New York Times or any other newspaper every day. More or less every day you can see stories relevant to energy and environment on the front page of the paper. We're organized along 4 distinct science programs. This is a relatively new organization for the JGI, even though we've had these core areas. And the reason for doing this is because the emphasis at JGI because of expansion of new technology sequencing that you'll hear about a little bit later, has focused the critical challenges for us are not getting the sequencing done. The critical challenges are actually getting the analyses done. And by having ourselves organized along these several scientific program lines, we can concentrate on analyses that investigators with specific kinds of projects require. So we have a plant program that's managed by Dan Roxar with help from Jeremy Schmutz and Jerry Tuscan. We have a fungal program coordinately managed by Scott Baker, who will be here a little later today. The program will be presented today by Igor Grigoriev, who manages the fungal and eukaryotic microbe annotation program at JGI. We have a prokaryotic isolate program that David Bruce, sitting in the second row here, manages. There's important additional scientific oversight of that program by Nikos Karpidis and Jonathan Eisen, who are co-managing our Genomic Encyclopedia of Bacteria and Archaea project that you'll hear about later in the meeting. And finally, we have a very active metagenome program that Phil Hugenholtz manages, along with Susanna Tringe and with Nikos. So these four science programs and these investigators are people that you may want to search out over the course of the next few days to understand a little bit more about what they do, and you'll be hearing from each of these programs shortly. We're very proud of the throughput of the JGI over the last several years. We were recently reviewed by OBER. They do that every 3 years. So in the course of that, we put together our throughput. We are now the world's largest producer of prokaryotic isolate genomes, bacterial and archaeal genomes, with almost 250 finished genomes produced in the last 3 years. We also are a leading producer of metagenomes, with more than 24 metagenomes produced and made publicly available over the last 3 years. And we've generated over 3 million ESTs in support of eukaryotic annotation projects, as well as providing gene space coverage for organisms that are too complex, using current methods for their genomes to be effectively tackled. We have a core group of JGI scientists, a relatively small group of JGI scientists that help us to carry out these projects and provide the critical path of the scientific mission for the JGI. And some of these investigators are here. I've put the rogues gallery up here on the right so you can begin to recognize them and hunt them down if you have individual questions. Dan Roxar, as I said, runs our plant genomics program, and he'll be talking a little bit later. Nikos Karpidis is next door running the Integrated Microbial Genomics Workshop and plays a critical role in pushing the envelope for microbial comparative genomics. Is an very active comparative genomics group at JGI. Phil Hugenholtz runs our metagenome program. He was a postdoc with Jill Banfield, got in on the ground floor of metagenome projects, and is currently driving the application of new technologies to metagenome projects. Len Pinocchio manages our technology development group, and along with Fong Chen, is trying to figure out how to use these new applications, these new sequencing technologies, for maximum effect for JGI sequencing projects. Cheryl Kerfeld is a recent hire who manages JGI's educational program, but also is a structural biologist and is developing a structural genomics focus at JGI. Jonathan Eisen, who I haven't put up here, has a primary point, who I haven't listed here, is actually plays a critical role, spends part-time at JGI in our microbial genome program. And Eddie Rubin has an active role in virtually everything that we do. My job is to herd all these cats and keep them on the rails. Well, we're a user facility, as I said, and I'd like to talk a little bit about what that means. What exactly is a user facility? This is a Department of Energy concept that grows out of the idea that DOE does big-scale science. DOE does things that individual investigators or universities or even groups of universities may not be able to afford, and that's why DOE was such an important player in getting the Human Genome Project off the ground. DOE runs lots of user user facilities. These are synchrotron light sources, colliders, big-scale science machines for physics. But at the end of the human genome era, it dawned on some wise folks at DOE that high-throughput sequencing for scientists who may not have access to high-throughput sequencing could really stimulate American science relative to DOE missions. And the concept was to make this high-throughput sequencing available to scientists, but not just the sequence, to also provide analysis, access to new technologies as they came along, the development of new strategies, and critically the access to JGI scientists so that good ideas from the community relevant to DOE missions could benefit maximally from the application of high-throughput DNA sequencing and analysis techniques. And that's really how the JGI's user facility came to be. It followed, as you see here, it followed the Human Genome Project with the initial formation of the DOE Microbial Genome Program. This program that Dan Drell ran until 2007 grew out of what Dan Roxar, I think, is appropriately called a JGI stunt, in which the JGI sequenced a handful of microbes in a single month. It nearly killed us. The quality of those genomes was not particularly high, but it did teach us a lot about what would be required to get DNA in the door and be able to maneuver a large number of projects through the doors and out into the community's hands in a relatively short period of time. That program ultimately grew and was very successful, but was incorporated into our current user program, the Community Sequencing Program, in 2007. The Community Sequencing Program was targeted on all DOE missions, not just microbes, and that program continues today. There was briefly what was called the Lab Science Program, which was a DOE National Lab specific sequencing program that's also been rolled into the Community Sequencing Program. And recently we've had, in 2009, the addition of new kids on the block. OBR and DOE have funded several bioenergy research centers that are focused in a Bell Labs kind of way on alternative fuels, and we provide the genomic infrastructure and sequencing for these three new bioenergy research centers. But just a word more about the Community Sequencing Program. This is a program that targets sequence projects of the highest scientific merit, relevant to DOE missions, as I described earlier. We carry out a peer review that's coming up in a couple of months, and ultimately this community sequencing program occupies about 50% of JGI's annual capacity. We've approved almost 200 projects from the bacterial and archaeal isolates that have been submitted over the years, 33 metagenomes. You can see the rest of the numbers here. Altogether, close to 300 projects have been approved and are either in the works or have been completed. And these, not infrequently, push the envelope of sequence-based science and end up in high-visibility journals. And I think it's the ability of JGI investigators to harness new technologies and new sequencing strategies in combination with scientists like yourselves who understand the relevant biology for these DOE mission-oriented projects, and it's that coming together of community scientists and JGI scientists that ultimately produces the best and highest visibility science. Our proposal process, we usually advertise the current call in November and December of each year. We have letters of intent that are due in January. Generally, those are screened for DOE relevance primarily, and 80 to 90% of letters of intent will be approved for subsequent full applications. Full applications were due yesterday, and we had 135 proposals submitted. Those go through technical review at JGI. Ultimately, an independent external peer review panel will review those, producing rank order of proposals that's then allocated for sequencing and analysis by our scientific advisory board in consultation with OBER. And the successful proposals are announced at the end of May or early June. So I encourage you, while it's late for this year, but keep your eyes open, check the web next November for our next call. Just a word more about the Bioenergy Research Centers. These are large-scale centers with missions in alternative energy. They are, by definition, JGI's top priority, but that doesn't mean that they compete directly with JGI's other users, with you all. And ultimately, we have the capacity to be able to meet both the needs of the bioenergy centers. The bioenergy centers currently are occupying less than 10% of our capacity in fiscal year 08. We'll expect it to be a little more in fiscal year 09. But there's plenty of room for these two, for the community sequencing program and the bioenergy centers to coexist. Over the last years, we've had a dramatic growth in the number of users with whom we interact. We have nearly 700 PIs and collaborators associated with individual sequencing projects. We're delighted with that. Ultimately, our goal is to have a user in every congressional district across the U.S., in case we need you. We also interact with people outside of JGI's direct sequencing projects. So we have annotators that are responsible for improving gene models for eukaryotic organisms. Igor Grigoriev manages that program and will be speaking to us a little bit later in the morning. The integrated microbial genome system now allows for organisms to be analyzed pre-publication through the IMG-ER. So if you've had an organism sequenced somewhere else and want to use the IMG for analysis, that can be done without us carrying out the sequencing. We have workshop participants, of which you all are participating today, but also IMG workshops, annotation workshops, other workshops through the year. And then we have a brand new educational program that Cheryl Kerfeld is managing that allows university professors to take JGI-created microbial genome data and have their students annotating real-live, first-pass microbial data. And for many students, this is a really seminal experience because it allows them to actually interact with real data, unlike the usual laboratory setting in undergraduate institutions where people are dealing with experiments where they know the answer. It's the first time that many students have to interact with real data. So we're pleased you're here. The user meeting promises to be very exciting. We have users from around the world that convene here for the next 3 days to hear about science, and importantly to interact with JGI's scientists and staff. So I encourage you to reach out. And a critical group of people that you also will need to know about if you're interacting with JGI at all are our project management organization. David Bruce, who's acting director of the Microbial Genome Program, also manages this group. These are the people who are responsible for getting your projects done, and they have the specific responsibilities that you've seen here. Carrie Berry manages our eukaryotic whole genome shotgun and metagenome projects. Krista Pinocchio manages our ESTs. Susanna, who will be talking about the metagenome program, manages our metagenome projects. And Lynn Goodwin manages all of our bacterial and archaeal isolate projects, along with David Sims at Los Alamos, and I know Lynn is here as well. So with that, I'll stop and we'll move on to the rest of the morning.